Welcome to the next segment of Chapter 14. We're going to be talking about predicting the outcomes of a mating and how Mendel developed his laws. Okay, so let's talk about how you can predict the results of a genetic cross or a mating between two individuals. So Punnett squares are actually used to display the cross and what the potential outcomes of a cross would be in order to predict the results of a mating between two individuals. And what I mean by results are the potential genotypes and, of course, the phenotypes if you know the heritable information, for instance, if it was dominant or recessive. Now, crossing individuals of known genotypes are the easiest. Now, let's look at an example of a cross here. So look, we have the mother on the top, across the top, and the father, so this would be the mother here, and then we have the father across the bottom. Now, what you do when you create a cross is to separate the gametes, okay? So this is a simple monohybrid cross. Now, how you separate the gametes is basically, remember, each of the alleles are going to be separated in gametes, so you basically just separate the alleles, right? So this mother is a heterozygote, so one capital A here and one capital A here. So these two represent eggs, right? So this is one egg and this is another egg. And then the father, again, is also heterozygote, so then you want to put the capital A in one sperm and the lowercase a in another sperm. So these represent the sperm. Now remember, fertilization is the crossing or fusing between an egg and sperm. So when you fuse this egg and this sperm, you end up with this right here. And then when you fuse these two right here, you end up with this. And then I'm going to go ahead and fill in this Punnett square. Okay, so we see that when you basically you're going to put the A from this egg right here in both of these boxes. Okay, so this one and this one. And then this capital A will go in this one and this one. Okay, and then this one, the little lowercase a, will go down here, and then down here, lowercase a from this one goes here and here. Okay, so now you have the results of this cross. So this allows you to predict what's the genotype and, of course, the phenotype that the next generation may have. So what this is saying is that in terms of genotype, okay, we have a one homozygous dominant, and there are two heterozygotes, okay, so to two heterozygotes, and then to one homozygous recessive. That's a 1 to 2 to 1 genotype ratio. Now, in terms of phenotype, okay, let's say this is the capital A is brown eyes, okay, and the lowercase a is blue eyes, okay. Then, so we have basically three individuals are going to have brown eyes, right? So because brown eyes is dominant, each of these three individuals have a capital A, so we have a 3, and then one individual will be blue eyes. So that'll be phenotype, PHE ratio. I'm going to fix that. Phenotype ratio. Okay, we're missing an N here. All right, so that's how you would do a cross. Look at the flowers. Okay, so if we did the flowers here, you would end up with, right, so we have the P here, capital P, and then lowercase p, Now, this kind of shows the problem with using a letter in which it looks the same, uppercase and lowercase. So this can start to get confusing if you don't make it significantly different. Often the lowercase p has a line over the top, okay? Sometimes it's written like this, right? So that's another way to do it, okay? So that's how you would do that. Of course, this is the result. So when we actually do this, we have capital P, lowercase p, then we have capital. Okay, so we'll bring down all the capitals. Okay, lowercase, 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 lowercase. And you can see the F1 generation are all heterozygotes. Okay, and this means they're all purple. Okay, so 100% and 100%. Another thing that he did was, or Mendel, that is, he took two of the F1 generation, two of the heterozygotes, and then did a cross here. So you can see the cross is already done. And this is where he came up with the 3-to-1 ratio of purple to white in terms of phenotype. So this is phenotype. Okay. The genotypic ratio, okay, so genotype, okay, is going to be a 1 to 2 to 1 ratio. So what we just did was a cross between two individuals in which we knew the genotype. Now, we're not going to always know the genotype, so we must do a test cross. So there are instances where we wouldn't know the genotype, and this is what I call the mystery genotype. Okay, so it's a mystery. It's a mystery because let's say you picked up a flower that was purple. How do you know what the genotype is? Okay, so it could be an individual that's homozygous dominant or a heterozygous individual. So what you want to do is do a test cross. And the test cross involves crossing this mystery individual. So we have the mystery guy. Okay, this person or individual can be homozygous dominant or a heterozygote. Okay, and you want to cross it with something that's known, right? So you want to cross it with a genotype that's actually known. And the only way you're going to know a particular genotype is if it has the recessive phenotype. Then, because you know that for an individual to express the recessive phenotype, both the alleles must be the recessive allele. Okay, so here I have two lowercase p's. Okay, so that's the instance where you would want to do a test cross. Now, after you do the test cross, what you want to determine is what the phenotype of all the offspring are. Okay, so if any of the offspring have the recessive phenotype, then the parent must have been a heterozygote, right? In order to produce two little p, right? So remember a recessive phenotype, okay? One of the parents must carry, both parents must carry one of the recessive alleles. Now, here we have the purple flower. It's the mystery individual. Okay, so that is our mystery individual. And remember, that individual can be uppercase P, or uppercase P, lowercase p, which represents the homozygous dominant or the heterozygote. And then you want to cross it with a known genotype. Okay, and in this case, we know the genotype of an individual that is expressing the recessive phenotype. Okay, so that would be the white flowers. So you know, you pick up a white flower, you know what that genotype is. You know that that individual has 2 chromosomes which have the recessive allele on each one. All right, so now you do the cross. So once you do the cross, let's say this is your outcome. Okay, so here, this is an experiment, right? So you have a prediction, and then these are the results right here, okay? And if all of the results, if all the flowers are purple, right? So then that individual had to have been the homozygous dominant. Now, here's your other option, right? So this is what also can happen. If any individuals, and it'll typically be about 50%, but if any of the recessive phenotypes appear in your offspring, then that individual had to be a heterozygote, that mystery individual. Okay, so that's what a test cross is meant to do. It's meant to determine the genotype of a mystery individual, and usually that individual is going to be expressing the dominant. Now let's talk about the law of segregation in relation to Mendel's work. And this is going to become important because we're going to be dealing with, rather than monohybrid crosses, dihybrid crosses. And what that means is we're looking at two characters rather than just one character. Now, Mendel derived the law of segregation by following just a single character. Okay, so here, remember the F1 offspring after mating to, alternative phenotypes of the parents. All of them were heterozygous. And then the cross between the F1, okay, which was the picking two of the heterozygotes from the F1 generation, which would result in the F2 generation, and we see the 3-to-1 ratio. Okay, so he derived that law of segregation from there. Now, the law of independent assortment was derived from crosses when he was investigating two characters. Okay, so here he investigated two characters at a time. This is called a dihybrid cross. Okay, so if you crossed true breeding, okay, which differ in their characteristic producing dihybrids, then the F1 generation are going to be heterozygous for both characters. Now, dihybrid cross, shown in your textbook, is one where the P color and shape were looked at. Okay, so a cross between the F1 dihybrids, okay, so both of them are heterozygous for both of the characters, it would result in a 9-to-3 to 3-to-1 ratio. And it's really representing different combinations of each of these characters, okay, or the traits associated with the characters. So we have P color. The P color can be green or yellow, and the P shape can be round or wrinkled. So let's go ahead and look at this cross. Okay, so we're gonna take it through this cross from the P generation through the F1 generation and all the way through the F2 generation. Okay, so looking at the P generation, we can see that we are crossing a yellow P and a green P. The yellow P has the round phenotype and the green P has the wrinkled phenotype. These are opposites phenotypes for both the characters. So here, of course, the genotype is going to have 4 letters, right? So the Y's represent the color and the R's represent the shape of the P. So here we have a double homozygous dominant individual crossing with a double homozygous recessive individual. Anytime you do a cross between two opposite phenotypes, even if you're looking at two characters and there's true dominance or complete dominance happening, the F1 generation are all going to be heterozygotes and they're going to display the dominant phenotype. Okay, so here we have the dominant phenotype in the F1 generation. However, the genotype looks different than any of the parents because remember, it's a combination of the parents. Right, so basically the genotype is going to be heterozygous for both of the characters. Now let me go back to the gametes. When we look at the gametes here, each of the gametes after the separation of the alleles, each of the gametes here are going to have the dominant Y and the dominant R. So it's not a single letter here, it's 2 letters because each of them are separated individually. Right, so into each gamete here, and there's only 2 options, right? So it's only a capital Y or a capital R. Here, same thing. Lowercase y and lowercase r will be in all the gametes of this individual. Now, Now we have the F1 generation. Now, if we want to go to the F2 generation, we're going to cross two of the F1 generation. In order to do that, you must separate the gametes into basically multiple combinations of the genotypes here. Okay, so what you must do is a foil. Okay, so I'm gonna do this foil here. Give me just a moment. Okay, so here the foil is actually shown in the boxes. Okay, so how did this? Remember what foil is? Okay, so it's first, outer, inner, last. Okay, so remember in algebra. So if you actually did this right here, okay, the first ones would be the capital Y and the capital R. Okay, so we have the first. And then the outer ones are going to be the capital Y and the lowercase r. Okay, and then the inner ones will be the ones on the inside. So that's a lowercase y and capital R. Okay, and then the last are going to be the last one in each of the parentheses. So we have the lowercase y and the lowercase r. Okay, so these are all the potential gametes in each of the two F1 generations. Now, to determine, basically, talking about this law of segregation, we have, considering the law of independent assortment, there are two hypotheses, right? So if these genes, right, did not segregate independently, right, so if they were not independently assorted during meiosis, then this would be the result. Okay, and what would happen is some of these would be transmitted from one generation to the next together. And then if they did independently assort from one another during meiosis, meaning that they were not linked genes, then you would get this result right here. Okay, so if we're looking at two characters, okay, and they did not independently assort, you would get a ratio that's very similar to looking at one character, right? Does that make sense? That they basically were transmitted or basically recombined during meiosis together. So then you would get something like a 3-to-1 ratio. Now, if they did independently assort, let's say they are not linked genes and they are far enough apart on the chromosome from each other that they would be separated during meiosis and crossing over, right? Then you would get a 9-to-3 to 3-to-1 ratio of phenotypes. And I'm going to do an example of this. It's better that I actually show you the example in a separate segment so that you can see me actually work out a problem, so that you can see me do the FOIL. So the law of independent assortment, again, using a dihybrid cross, this is how Mendel basically developed this particular law. And the law of independent assortment actually states that each of the pairs of alleles segregate independently of each other during the formation of gametes or during meiosis. This law applies to genes that are on different chromosomes that are non-homologous. Okay, so they're different. Let's say non-homologous, meaning they're genes that are on, or basically alleles with the genes that are on chromosome 3 and 5, right? Two separate chromosomes, and that are not homologous chromosomes ultimately. And they must be far enough apart on the chromosome to actually have a higher chance of being segregated. So if they're closer together on the chromosome, there's a much lower chance of them being separated. Now, genes that are located near each other on the same chromosome usually are inherited together. So sometimes you see similar characteristics in individuals with certain characteristics. For instance, often red-haired individuals have freckles as well. Right, so that would be an example. Now, I am not personally sure whether those genes are located near each other on the chromosome, but when you see traits that are together, that they seem to be inherited together, that means they're probably very close on the chromosomes so that they did not get segregated and there was no variation. Okay, so this concludes this segment. I kind of talked a little bit about Punnett squares, although there will be another a couple segments with examples on how to work out problems using a Punnett square. And then I also talked about Mendels Laws and how he developed the law of segregation using monohybrid crosses and the law of independent assortment using the dihybrid crosses.